Eukaryotic cell. The cell is the basic structural, functional, and biological unit of all known living organisms. Cells are the smallest units of life. The eukaryotic cell is enclosed by a membrane that separates the interior from the outside environment. Inside the cell, enclosed by another membrane, lies the nucleus that holds the cell's DNA. In between these two membranes lies the cytoplasm. The cytoplasm is a thick, gel-like substance and is made up of roughly 80% water and is usually clear and colorless. It serves as a molecular soup, and it is in the cytoplasm where all the cellular organelles reside, the organs of the cell. Eukaryotic cell. Eukaryotic cell. The cell is the basic structural nuclear pore. Nuclear pores allow the transport of molecules across the nuclear pore. Nuclear pores allow the transport of molecules across the nuclear envelope. This transport includes RNA moving from the nucleus to the cytoplasm and proteins moving into the nucleus. It is notable that a single nuclear pore can actively conduct 1,000 translocations per second. Nuclear pore. Nuclear pores Ribosome. The ribosome is a complex molecular machine found within all living cells that serves as the site of biological protein synthesis. Ribosomes link amino acids together in the order specified by messenger RNA molecules. Ribosomes consist of two major components: the small ribosomal subunits, which read the RNA, and the large subunits, which join amino acids together. The ribosomes and associated molecules are also known as the translational apparatus. Ribosome. The ribosome is a complex molecular nuclear pore. Nuclear pores allow the transport of molecules across the nuclear envelope. This transport includes RNA moving from the nuclear pore. Ribosome. Nuclear pore. Nuclear pores allow the transport of molecules across the nuclear envelope. This eukaryotic cell. The cell is The 2017 Nobel Prize in Physiology or Medicine was awarded to Jeffrey C. Hall, Michael Rosbash, and Michael W. Young for their discoveries of molecular mechanisms that control the circadian rhythm. Life on Earth is adapted to the rotation of our planet. For many years, we have known that living organisms, including humans, have an internal biological clock that helps them anticipate and adapt to the regular rhythm of the day, the so-called circadian rhythm. The clock regulates critical functions such as behavior, hormone levels, sleep, body temperature, and metabolism. But how does this clock actually work? The 2017 Nobel laureates were able to peer inside our biological clock and elucidate its inner workings. Their discoveries explain how plants, animals, and humans adapt their biological rhythm so that it's synchronized with the Earth's revolutions. During the 1970s, Seymour Benzer and his student Ronald Konopka asked whether it would be possible to identify the genes that control the circadian rhythm in fruit flies. Both humans and fruit flies carry their genetic material in a distinct nucleus, located within each of their cells. Because of this common denominator, the fruit fly, having a very basic level of physiology, is used as a model organism in studies that are applied towards deciphering more complex processes, such as those found in humans. The cell is the basic unit of all living organisms. Cells are the smallest units of life, and are often called the building blocks of life. The cell membrane is the outer lining of the cell. It is a biological membrane that separates the interior of the cell from the outside environment. The basic function of the cell membrane is to protect the cell from its surroundings. Enclosed by the cell membrane is the cytoplasm. It is made up of a jelly-like fluid and other structures that surround the cell nucleus. The nucleus is a structure that contains the cell's genes. The nucleus is also surrounded by a membrane called the nuclear envelope. Studying the fruit fly, Benzer and Konopka demonstrated that a gene could influence the circadian clock, and they named this unknown gene Period. The question was how Period could actually achieve this influence. The 2017 Nobel laureates Jeffrey C. Hall, Michael Rosbash, and Michael W. Young, who were also studying fruit flies, aimed to discover how the clock actually works. In 1984, they succeeded in discovering the period gene. 10 years later, in 1994, Michael Young identified a second gene involved in the circadian rhythm: timeless. The period and timeless genes produce mRNA molecules that carry genetic information. The mRNA is transported from the cell nucleus to the ribosomes in the cell's cytoplasm for the production of Per and Tim proteins. A ribosome functions as a micromachine for the making of proteins, where the mRNA serves as a recipe. Jeffrey Hall and Michael Rosbash reasoned that the Per protein enters the cell's nucleus and blocks the period gene activity. But it was not clear how Per entered the nucleus. In elegant work, Michael Young showed that when Tim bound to Per, the two proteins were able to enter the cell nucleus. Once inside the cell's nucleus, Per and Tim prevent their own production by blocking the period and timeless gene activity. The Per and Tim proteins that remain start to degrade. Because of this, the period and timeless genes become active and once again allow production of mRNA that generates Per and Tim protein. One cycle of turning on and off the genes takes about 24 hours, and this is the clock that generates the circadian rhythm.